experiments. I don't actually know if he got the Nobel Prize for them, but they're famous classical experiments developed in biology. And he was working on embryos at a little bit later stage than Hilda was, to try to understand more about this neural patterning event in the embryo. And so he was taking same sort of idea, cut out little pieces of tissue and transplant them over to a host embryo and see what happens, and see if you can make some sort of conclusion about signals and where they're coming from and what they're doing for the embryo. And so what he's done here is to take different areas of the embryo as gastrulation is proceeding. So on the top there in A, he's taken the tissue that has gone in first and migrated the furthest within the embryo, cut that little chunk out. It's a mixture of both ectoderm on the outside and mesoderm on the bottom, so the gray and the red there. Transplant that over to a early gastral embryo, just stuck it right into the blastocyst cavity there, and see what happens. And amazingly, these transplanted tissues, they survived and they built some extraneous tissues in the embryos that developed from this. And depending on what region was transplanted, you got this series of different structures that were progressively very anterior to slightly less, and then more and more posterior in character. So this very first tip of the piece, that gave the structures that are highly, highly anterior. So forebrain is really right at the anterior tip. Balancers are these little structures on the side of the embryo that help it maintain its balance. And mouth, also very anterior. And then taking a slightly later involuting or more posteriorly localized piece, now you've got some kind of intermediate stages like midbrain, eye, and ear. And then the most recently involuted, these gave the trunk and tail, which are the pieces with the most posterior character or self-fate to them. So obviously there's some regionality here that's happening. And it has to do with neural induction. So once again, it's not just that we get any neural induction, but that neural induction has a character to it of being more anterior in nature or more posterior in nature. And there may be some different signals, possibly a different morphogen gradient, we don't know, but some kind of different information from this involuted tissue that's beginning to pattern the embryo as gastrulation proceeds. And it's probably doing it by an induction process. So here's kind of a summary of this. So 1, 2, 3, 4, 5. This means what tissue went in first, and then followed, and then followed, and followed. Number 1 went in first and got the furthest. And that's going to be the most anterior in character. So if there's induction occurring here, those cells have induced that signal in a way that produces a most anterior cell fate. Okay. You can also think of this in terms of timing. So if we take the same region, dorsal lip of the blastophore, but at different times, really, really early when gastrulation just began, or kind of later after gastrulation had been going for a while, and you do this transplant experiment, you get out the most anterior fates for the earliest involuted tissue. And that actually makes sense with the previous experiment. It was the same actual cells that would have gone in first that had the most anterior character, and then if we took them either right when they were going in or after they'd gone in and progressed a while, they still have that very anterior inducing ability. Okay, so this is beginning to put this together. So here's showing you the model for anterior-posterior induction that's starting from signals in the dorsal blastophore lip and seeing these being spread out across the embryo, and some inductive signals going from mesoderm to the overlying ectoderm. And there are a number of different players here. So in green we have the BMP inhibitors. So remember I said there'd be a group of paracrine factors on the roof of the archeneron that's keeping a stripe free from BMPs. These are shown here in green, and they're going to inhibit BMPs and keep that for a neural cell fate. In purple, this is another factor. It's actually in a gradient. It's retinoic acid. Let's see. Oh, actually, sorry, Wnt. And you're going to see higher Wnt in the later involuting tissues and less Wnt in the first involuting tissues. So this is a signal. We already heard Wnt was important, but now it's going to be reused in this anterior-posterior characterization as well. So we'll have an area of Wnt inhibitors that are keeping a Wnt-free area right here in this yellow. And that gives a certain neural fate that involves brain and pharynx. So let's see, did I... Okay. All right, so I think I talked about both of these. We have a BMP-free stripe, and also we have a Wnt gradient where we'll have high Wnt versus low Wnt, and both of these are involved in establishing regional polarity and establishing an area where you're going to have a BMP-free region to become neural in character. Okay, so this summarizes into kind of a two-gradient model for establishing anterior-posterior patterning and for neural induction. And so over here on the more posterior side, you'll see high Wnt, high retinoic acid, and then low on the other side. And then green means high BMPs in the non-neural areas, and then low BMPs where you're going to get neural tissue development. Okay. So, and then this is contrasting mesoderm induction and neural induction, and kind of showing you that some of the mechanisms are in common. So both of these events, which happen at different times for the embryo and do different but similar things, they're both involving induction, and they're both involving regional polarity. So I think you can start to get a sense that there's some themes here. There's some mechanisms that embryos use to accomplish certain goals. Like we want to choose one fate versus the other. We want to choose within that fate different characters. Here's how you can do it. You might apply a morphogen. You might apply a paracrine factor. Okay. So within this embryo, as some of this mesoderm tissue is migrating in, if we take those cells that are exactly on the midline of the embryo, moving from the dorsal side to the ventral side, if we consider just those very central cells, they're going to develop into a tissue known as the notochord. And so here is, this top one is showing you a cross-section through the embryo in the plane that we've been looking at, so dorsal to ventral. Or if you flip that 90 degrees and do a transverse section, here's that little group of cells right exactly in the midline. These have become the notochord. And the notochord is kind of a stiff rod that runs from anterior to posterior in the embryo. And it is a transient structure, and since it's stiff, it can give the embryo some shape to it, but it's not going to actually persist and become a backbone later in adult life. These cells will degrade before that happens. But it is really important for patterning the nervous system within this embryo. So that's introduction to the notochord here. And then meanwhile, what's happening to the shape of this embryo? So as gastrulation is happening interior, this starts to reach its conclusion. Now we see some movements of the cells on the ectoderm or outer side of the embryo. And on the outside of the embryo, over that plate where the archeneron is, we see a kind of a flattened area. And this is called the neural plate. And again, that term plate is used for a kind of a flat area within the embryo. And on the sides of this neural plate, we see cells kind of moving laterally and bunching up to form these two ridges. So as these ridges form, they're going to move more and more towards the midline of the embryo. So two ridges of cells, and then they just literally move their location towards the center of the embryo. And here you see these two ridges that are meeting, and they're eventually going to fuse together. And so this is the process of the next major morphological feature within the embryo. It's called neurulation. And we're going to form here a neural tube, which will eventually become the brain and the spinal cord. So here's looking at those events in a cross section, and up here at the top you see in green there the neural plate, which is the flattened region, and then on either side is kind of two areas that will become the little ridges called neural ridges. And this group of tissue, this flat neural plate, is actually going to buckle inward. So here you can see it forms a groove, and this runs along the embryo anterior to posterior. And meanwhile, the two neural ridges are folding up and moving toward the midline. So we're going to basically fold in a little trough of cells and then seal it off at the top. And this makes a tube sitting right there on the top of the embryo. And eventually this will seal over so it's flat on the top, and then we have this tube running along the length of the embryo. Does that make sense? How that's, sometimes I never know. I try to explain this. So we get a tube. And there's also, I'll just point them out for now because they're a really interesting group of cells that we'll come back to. There's a few groups of cells here that leave the sheet-like epithelial arrangement of cells and become just loose mesenchymal cells. And they're hanging out right here between the overlying ectoderm and this neural tube. These loose cells are called neural crest cells. They're going to migrate all over the place in the embryo and take on all kinds of different cell fates. So they're sort of unique and fascinating for their own right. But for right now, what we have is ectoderm now on the outside, a tube running from head to tail, and below that, in the mesoderm portion of the embryo, these cells that we're bisecting right at the top of that are archeneron, and these will form that rod-shaped structure known as the notochord. So these three together, here you can see it in an embryo, a distinctly tube-shaped thing. Underneath that, a bar-shaped thing that's the notochord. And then the remnants of the yolky, big gut cells now in the middle of the embryo. Okay, so this process of forming a tube, the tube is called the neural tube. It's going to become the brain and spinal cord. And this basically happens for all vertebrates. So whether you're talking fish or mouse or human, it all happens. There's some difference in the way that the tube seals shut. So in humans, you kind of get these ridges moving up and meeting at different places, and then it seals in between those two, and becomes eventually a full-length tube along the anterior posterior. In chicken, it kind of starts at one end and then just zips shut as the neural tube forms along that anterior. So those differences are not really functionally that important as time goes by. So neural tube defects, actually one of the most common birth defects out there. If this doesn't happen correctly, the embryo will die. If it happens mostly correctly, but not quite perfectly, you can get some really severe birth defects out of this, some of which are compatible with survival, in other cases not. So here you see the two neural ridges are meeting and fusing. Those little circular / square shaped things, those are somites, which are just chunks of mesodermal tissue that we can recognize as the embryo develops. So here is two names for some common neural tube defects. One is anencephaly. So this is referring to head cephaly. An is a negative prefix. And what's happened here is the neural tube has formed pretty much fine in the truncate and posterior, trunk and tail, but not in the head. So it remained open in the head due to some kind of mispatterning event or some kind of problem, maybe in a gene. And therefore, the brain development was really severely inhibited. And if this is too far gone, it's not going to survive. Okay. You can also get in humans kind of the opposite scenario where the head portion of the neural tube forms, the trunk does, but right down at the most posterior region, there might be a little place where those two ridges fail to seal. And then that leaves kind of this open, splayed out neural tube trying to form ectoderm and spinal cord right down here at the bottom. And so this can give a condition called spina bifida, and sometimes this could be surgically corrected depending on how severe it is and the person could survive. But I show these to sort of bring in the patterning events and the morphological events that are happening right now. I think that's our last slide. Yeah. Any questions?